Advances in Dementia Research webinar. My name is Julie Wong. I am the Senior Manager of Education and Community Programs at the Alzheimer's Society of Toronto. A little bit of housekeeping. I do ask that you place yourself on mute during the whole presentation. We are recording this webinar, so it will be available on both the Alzheimer's Society of Toronto YouTube channel, as well as on the TDRA website. If you do have questions, you can feel free to place them into the chat, and we'll get to as many as we can after the webinar. So this Advances in Dementia Research Series is presented by the Alzheimer's Society of Toronto in partnership with the Toronto Dementia Research Alliance. These series discusses recent updates in dementia research and highlights dementia research studies that are occurring across Toronto. Today, our speaker is Dr. Marcellus, and Brittany will be introducing him shortly. For those who are not necessarily familiar with the Alzheimer's Society of Toronto, I did want to give you a bit of an introduction. AST is our short form. We provide information, education, and support to people living with dementia and their families and friends, healthcare professionals, and the general public within the M postal code. We service all dementias, and a diagnosis is not required to access any of our programs and services. Our services are offered in person, as well as virtually through Zoom and the ALS Educate e-learning platform. To learn more about what we have to offer, please visit our website at www.alz.to and take a look at our calendar of events for upcoming offerings. I'd like to introduce you to our partner, the TDRA, or Toronto Dementia Research Alliance. Established in 2012, the TDRA is a collaboration among the University of Toronto, Baycrest, the Centre for Addiction and Mental Health, Ontario Shores Centre for Mental Health Sciences, Sunnybrook, Unity Health Toronto, and the University Health Network. Together with its partners, TDRA aims to strengthen the link between basic science and clinical research to better understand, prevent, and treat dementia. So I'd like to hand it now to my co-host from TDRA, Brittany.
Thank you, Julie. So we can go to the next slide. Perfect. So AST and TDRA partnered on an initiative to make research more accessible to everyone. The Toronto Dementia Network, or TDN, is a website that lists dementia-related programs and services across Toronto. On this website, we created a section called Research Studies, which lists dementia studies that are open for participation across the city. Each study listed is led by TDRA-affiliated scientists and has been approved by a research ethics board. There are studies for people living with Alzheimer's disease and other types of dementia, mild cognitive impairment, and those at risk of developing dementia, as well as healthy volunteers and caregivers. The studies are all summarized in plain language, and to get started, the website link is shown here in orange. Information about the TDN can also be found on both the AST and TDRA websites. Next slide, please. Thank you. On the website, there are two main pathways. First, you can browse for a study using search terms, filters, and quick searches. When you find a study you're interested in, you can fill out the contact form to be connected with the research team, who will enroll you in the study if you're eligible. Or at the bottom here, if you're not sure where to start, you can fill out a questionnaire to be matched to a study. Any information you enter will be securely stored and protected. Next slide, please. Thank you. So this is what the website looks like. When you're on the page, if you're looking for something specific, for example, Alzheimer's disease studies, you can type it into the search bar along the top. You can hit submit with that term or apply more filters like type of study, age group, target population, or the study location, and then hit submit. Alternatively, you can use the quick search buttons we have in the blue boxes. If you'd rather fill out a questionnaire to be matched to a study, you can click the Match Me to a Study quick search box, or scroll to the bottom of the page and you'll see this orange button that says Can't Find a Study You're Interested in, which will also take you to the questionnaire. Each study summary, as shown in the box on the left for the Gen5 study, describes what the study is about, who can participate, the location, and the time requirement, all in plain language. The studies being discussed today are listed on TDN, so you will be able to read more about them and connect to the study teams there. Next slide, please. Thank you. And now I'd like to introduce our speaker. Before I do, we'll just send a reminder to keep any questions till the end of the webinar for our question and answer period if you want to unmute after. But do feel free to type them in the chat throughout and we'll keep track of them. We're grateful today to be joined by Dr. Mario Macelis. Dr. Macelis obtained his MSc in pharmacology, his MD, and his board-certified neurologist, all at the University of Toronto. He completed a clinical fellowship in cognitive and movement disorders and obtained a PhD in clinical neurosciences. He is a clinician scientist at Sunnybrook Health Sciences Centre and an associate professor of medicine at the University of Toronto. His research focuses on understanding shared genetic risk for neurodegenerative diseases of aging using genomic, cognitive, and neuroimaging data. He is the Canadian primary investigator for the Gen5 or Gen5, I'm not sure if I'm saying that acronym properly, I just say it in my mind all the time, but Gen5 project examining pre-symptomatic biomarkers of genetic frontotemporal dementia, and is the site primary investigator for what I called the DIAN-2, hopefully I'm saying that right, project. Both of these studies will be discussed further in this presentation. So thank you, Dr. Macelis, for being here.
Great. Thank you very much, Brittany and Julie, for giving me the opportunity to speak to everyone here today. And thanks, everyone, for participating. I'm just going to share my screen. Okay, I just want to double check with Brittany and Julie whether or not they see my presentation format screen.
I do,
Yep.
yes.
Excellent. And everyone can hear me okay? Great. Okay, so thanks for everyone who's attending. Thanks to everyone who's attending today. So I'm going to be talking about genetics in dementia diagnosis, and then I'll talk a little bit about current and future perspectives. So the audience is quite diverse, from family members, caregivers, through to healthcare professionals. So I'll make sure we leave ample time to answer any questions at the end. So the objectives of the talk is that we'll explain the role of genetics in different types of dementia, including Alzheimer's and frontotemporal dementia. And then I'll also describe two studies: the Dominantly Inherited Alzheimer's Network Trials Unit, that's the DIAN-TU acronym, and the Genetic Frontotemporal Dementia Initiative, or GenFI. GenFI is perfectly fine. I think to start off, I want to remind people that dementia is an umbrella term. And what I mean by that is that dementia just means having cognitive cognitive problems, which includes memory, but can also include other domains of thinking patterns that impairs an individual's activities and activities of daily living. Under this umbrella of dementia, there are many different subtypes of dementia, with Alzheimer's disease being one that we'll talk about today, and a second form is frontotemporal dementia. So not only will I talk about the genetics of these disorders, but I will explain a little bit about how they clinically present so that people can become attuned to the differences between these important types of dementia, both of which, cause immense suffering to both patients and their caregivers. So we'll first move in and talk about Alzheimer's disease. So the most common presentation of Alzheimer's disease is having a memory impairment, or what we call an amnestic syndrome, where memory is kind of the most predominantly affected domain. Other areas of function, such as language, may also be impaired, and medical term for language problems is aphasia, as well as executive problems, such as troubles with multitasking, being able to multitask, being able to carry out a sequence, bring things to completion, is another area that's affected in Alzheimer's disease. So in order to be diagnosed with Alzheimer's disease, you typically have to have most commonly a memory impairment, but other domains of cognitive function can also be affected. This has to be significant enough to cause a loss of social or occupational function, and also represent a decline from previous level of function. I want to emphasize here is that while Alzheimer's disease is the most prevalent and common form of dementia, less than 1% of Alzheimer's cases have a very strong genetic inheritance pattern. And the pattern that I'm discussing here is what's called autosomal dominant. I want to remind people that when we inherit our genetic background from our parents, we get one copy of the background from our mother and another copy from our father. And all you need to have is one abnormal copy of a gene in order to have what we call an autosomal dominant pattern of inheritance. When we contrast this with a different type of inheritance pattern, which is called recessive, you have to have two copies, one from your mother and one from your father, that are both abnormal in order for it to cause the disease. So I want to remind people that Alzheimer's disease, while it's the most prevalent form of dementia, strongly genetic forms of Alzheimer's disease are very uncommon, occurs in less than 1% of all Alzheimer's cases. And I'll give you some more background about this. Before I go on, though, I do want to say that the most common form of Alzheimer's disease is what we call late onset forms, so after the age of 65. And these are also what we call sporadic in nature. And what that means is that we don't have a strong genetic pattern to inheritance that's known. Just to remind people, if someone passes away with a diagnosis of Alzheimer's disease, and if indeed they do have Alzheimer's changes in their brain, this is what we see. We see these extracellular, so existing outside of cells, called amyloid plaques, beta amyloid plaques. And then within brain cells or neurons, we see these tangles, which are made up of a protein called tau. And so my mentor, Sandra Black, always told me that it takes at least two to tangle, and that includes the plaques and tangles of Alzheimer's disease. She'll also remind us that many people, as they get older, also have vascular changes in the brain, such as hardening of the arteries, or hardening of the veins, and these can also contribute to cognitive decline in late life. So when we talk about Alzheimer's disease, I'm not going to go through this slide in detail, but I'm just going to highlight that there are, can everyone see my cursor moving on the background? Yeah, I just wanted to confirm that. That generally it's been thought, and it's not completely accepted, that the first part of the process is the accumulation of these amyloid plaques by a protein called beta amyloid. And this sets the stage for multiple lines of damage that can happen to the brain, which includes inflammation and aggregation of beta amyloid into these plaques. The next step is thought to involve tau, where tau then accumulates inside brain cells. And then when tau accumulates inside brain cells in a pathological way, these brain cells die. And when these brain cells die, you get cell death and you get dysfunction of chemicals in the brain called neurotransmitters. And this is what results in part to the memory problems and behavioral issues that can arise. In order to get Alzheimer's disease pathology in the brain, and once again, I'm not talking about the clinical syndrome of Alzheimer's disease, but I'm talking about when we're able to look at the brain after someone has passed away and confirm that they have Alzheimer's in the brain, there are two pathways that are thought to lead to the final diagnosis of Alzheimer's disease on autopsy. One is where we have is a pathway that we want to promote, which is called the non-amyloidogenic pathway. And what this means is that this is a pathway that leads to less amyloid in the brain and a lack of accumulation in the brain. But there's another pathway, which is amyloidogenic, which means that beta amyloid is promoted to stick together and clump together, which results in these plaques, which sets the stage for later neurodegeneration. And this is the bad pathway. So when we're trying to develop treatments for Alzheimer's disease, we want treatments that will facilitate this non-amyloidogenic pathway and also reduce or prevent this amyloidogenic pathway. Now, there are a number of genes that have been discovered for Alzheimer's disease over many, many years. And all of them essentially are associated with the accumulation of amyloid in the brain. So having these gene abnormalities or gene variants or gene polymorphisms or mutations are thought to contribute to amyloid accumulation in the brain. So what I want to show here first is that there's presenile or strongly familial forms of Alzheimer's disease. This is what we term as genetic Alzheimer's disease that runs in families. And then there's the later onset forms of Alzheimer's disease, which can be both sporadic, but sometimes can also run in families. There are a number of genes that have been discovered that either will cause Alzheimer's disease if you inherit the gene or increases your risk. And I'll try to explain a little bit more about this. So the presenilin-1 gene is one of them. The presenilin-2 gene is another. And the amyloid precursor protein is another one of these genetic genes that are involved in the accumulation of amyloid in the brain and thus causing Alzheimer's disease. In these genes, we find mutations, which are very, very rare variations with a frequency in the population less than 1%. These, if you inherit one of these mutations, you will inevitably get Alzheimer's disease at a certain point. So once again, early onset genetic forms of Alzheimer's disease, if you have one of these mutations in one of these genes, you will get the disease, and there's a 50% chance of inheriting this disease because it would have been passed down from one of your parents. On the contrary, in late onset sporadic forms of Alzheimer's disease, there's another gene called the apolipoprotein E gene. And there's an allele called the E4 allele or epsilon 4 allele. I'll just say E4 because it's easier to see. If you inherit one copy or two copies of this gene defect, and this is a common variation. So, about 10 to 15% of people, have one copy of this ApoE4 allele. And it's much higher in the Alzheimer's population, meaning that it increases your risk for developing Alzheimer's disease. So if you inherit one or two copies, your risk for developing Alzheimer's disease goes up. If you inherit two copies, then you have about a 10 to 15-fold increased risk of developing Alzheimer's disease. The difference between this gene, it is a common variant. If you inherit it, it does not guarantee you will get Alzheimer's disease. It just increases your risk, and it can do so substantially. But it's important to know that it's not 100% causative, unlike mutations in these other three genes, presenilin-1, presenilin-2, or APP. So it's a little bit more background on the genetics of Alzheimer's disease. So we have these rare mutations that can cause it, and we have these common genetic variants that increase your risk substantially, but don't necessarily cause it in every person. So something important to keep in mind when we talk about genetic forms of Alzheimer's disease. So just to give you some more background, when we talk about early onset familial Alzheimer's disease, which are due to genetic defects, we see that the most common one of the mutations is presenilin-1 versus presenilin-2 versus APP mutations. In terms of the age at onset, presenilin 1 mutations, the age at onset is usually in the early 40s, but can range up to between 24 to 65 years old. Presenilin 2 mutations often can cause Alzheimer's disease between the ages of 39 to 75, with most of them still being in this earlier spectrum. And then these APP mutations are usually in individuals who are in their 40s to 50s, but sometimes can be as high as 60s. So important to keep in mind that this age of onset with these genetic mutations is much younger than the late onset sporadic form, which is due to ApoE allele, ApoE4 gene variants, and this subtype occurs typically after the age of 65 when you have the ApoE4. And if you have this variant in ApoE4, it doesn't mean with 100% certainty you will get the disease. This is the structure of the ApoE gene, and I just wanted to show that there are three fairly common genetic variants. One is called the epsilon-2 or E2 allele, the other is E3 and the E4. E2 allele is thought to be protective against Alzheimer's disease, whereas the E3 allele doesn't protect, nor does it increase your risk for it. However, the E4 allele increases your risk. There have been other genetic variants and mutations in, for example, the TREM2 gene that can also increase your risk for developing Alzheimer's disease. And there have also been protective mutations that have been found in the amyloid precursor protein, the APP gene that I mentioned earlier, that can also protect against cognitive decline. So when we think about mutations and common variants, we have to think about not only risk variants or causative variants, we also have to think that there may be protective variants that can protect against the disease. And this helps us when we're trying to develop new treatments that can follow suit on some of these protective genetic variants. Now I'm going to shift and talk about frontotemporal dementia, which is a different form of dementia. So frontotemporal dementia, in patients under the age of 65, it's the second most common cause of dementia after Alzheimer's disease. The age at onset is typically 45 to 65 years old. Patients present with predominant frontal and/or temporal lobe symptoms. And what this translates into, people can present with complex, inappropriate, stereotypic behavior and personality changes where they'll do things that are out of character. So rather than having a primary memory difficulty, patients may present with frontotemporal dementia by doing inappropriate things like going outside without their clothes on. They may start shoplifting. They may become argumentative and challenge people. Another variant of frontotemporal dementia is that they can also present with primary language difficulties where they can't find words, like it's very hard to put together a sentence. So that's one form of language variant. Or they can have another form of language presentation whereby they lose the individual meaning of words. And so it becomes difficult for them to comprehend when someone's speaking to them, okay? So contrast this to Alzheimer's disease, which in most cases presents with memory problems. Frontotemporal dementia presents with either extreme behavior and personality changes, or can present with more language problems in contrast to Alzheimer's disease. I want to keep in mind also, I also want to raise the issue is that, remember, these are neurodegenerative diseases, so that over time, other symptoms emerge. So someone presenting with behavioral features early on can later on develop both language and memory problems. And similar in Alzheimer's disease, someone presenting with a memory problem later on in the course of disease can present with language or behavioral issues. So as the disease progresses, while these diseases are can be distinguished early on in the clinical course, by the time they're at their late stages, they all start to look the same because ultimately all of the brain becomes affected by these diseases. Okay, so something to keep in mind when we're seeing these diseases, and why someone who has a diagnosis of Alzheimer's disease may, at one point during the course of the disease, have language or behavioral issues in addition to their memory issues, which started it. So I'm just going to keep on moving on. Patients also with frontotemporal dementia can also present with Lou Gehrig's features or ALS, and they also can have Parkinsonian findings on their exam as well. So just another clinical presentation. Now, contrast frontotemporal dementia to Alzheimer's disease. Rather than the 1% being strongly genetic in Alzheimer's disease, up to 30 to 40% of cases of frontotemporal dementia are strongly genetic, where there are inherited mutations. Okay, so keep that in mind, that this is much more of a strongly genetic disease than Alzheimer's disease. This is the picture of the brain of someone who presented with frontotemporal dementia in life. And this person presented with behavioral language problems. And you can see here, this is the parietal area of the brain and the occipital area. So these are areas that are involved in processing the visual environment. And this is showing the normal structure of the brain, where this part of the brain is nice and thick, and these are called gyri. If you look in the frontal part of the brain, you can see how it's kind of almost shriveled up like a raisin. So this is what we call atrophy or shrinkage of the brain. And so we see this selectively in the frontal lobes and the temporal lobes of the brain. And this is the pattern we see in frontotemporal dementia. There are two major changes that happen in the brain of people with frontotemporal dementia, at least the two most common ones. Similar to Alzheimer's disease, which is caused by the accumulation of two proteins, amyloid and tau, in the brain, frontotemporal dementia is caused by the accumulation of tau in the brain in some cases. And this was found to be inherited in an autosomal dominant fashion, meaning you just need one copy of this particular gene called the MAPT gene, which is here described as MAPT, M-A-P-T. This particular gene can cause forms of frontotemporal dementia associated with tau, inclusions called PIC bodies. And this is where in the old days, frontotemporal dementia used to be called PIC bodies. So once again, the first mutation that caused frontotemporal dementia, that was the first mutations that caused forms of frontotemporal dementia associated with tau accumulation in the brain, were due to mutations in this particular gene called MAPT. What was very interesting, though, when we studied these families, there was a bit of a genetic puzzle that emerged. So we found that there were patients, there were family members from families that clearly showed a strong inheritance pattern for this form of frontotemporal dementia. And it was in this area where the MAPT gene was located. But when we analyzed them pathologically, they had no tau protein in their brain. They had a different form of a protein called TDP43 in the brain. And what was found, there was another gene that was found in this area called progranulin. So this is another gene mutation that's found in the same area where the MAPT gene mutation has been discovered, that causes frontotemporal dementia. And these individuals that had progranulin mutations had a different protein that accumulated in the brain that caused their neurodegenerative form of frontotemporal dementia. And this protein was called TDP43. So what they did was they were able to map this area and identify the progranulin mutations as a cause for this. And this is what progranulin looks like in the brain. So we don't see those PIC bodies that I showed you in the previous cell, but what we can see are these kinds of cat's eyes type of inclusion that stain avidly for TDP43, which is another protein. Another story emerged whereby they found other families that had this TDP pathology, but they weren't associated with these progranulin mutations. And over years of study, they found this repeat expansion in a gene called C9orf72 as a cause for frontotemporal dementia associated with TDP pathology. But these patients also could have a form of Lou Gehrig's disease or ALS. And this is what the pathology looks like here. So there have been many other genes that can cause forms of frontotemporal dementia, and I won't go through them. I'd like to shift gears and start to talk a little bit about genetic testing. This applies to both our conversations about Alzheimer's disease and also about frontotemporal dementia. And so we have to develop an approach to Alzheimer's and frontotemporal dementia genetic testing. So when we talk about genetic testing, we want to make sure that we have our best possible and clearest clinical diagnosis as possible of either FTD or Alzheimer's disease is the correct one. We do this, we also want to take a detailed family history to ensure that we have this type of autosomal dominant pattern where it seems like a member of every single generation has this particular disease, either Alzheimer's disease or frontotemporal dementia. Then patients and or family members can be referred to a clinical geneticist or a genetic counselor. And then after discussion with the index case being someone who is affected with the disease, as well as their caregiver, you want to obtain consent to get genetic testing in the index case. And then the blood is sent to test a bunch of genetics causes in certified genetics labs. And then we get the results of these and they're communicated to the patient and their caregiver. If positive, this opens up the possibility for what pre-symptomatic genetic testing, and this can be offered to relatives, but only after a thorough discussion. Why? Because knowing your genetic mutation before you develop symptoms of the disease can be quite stressful. If you know you have the mutation, then it can cause anxiety. It can cause depression. If a member of your family doesn't have the mutation, then those members can feel guilty because they didn't inherit it when they know that their brother or sister did. So there's a lot of implications about finding genetic results. In addition, finding out genetic results, and luckily in Canada, we have a genetics protection law. But if an insurance company, for example, somehow found out your genetic result, they can deny you insurance. So it's something to keep in mind, whereby when we disclose results to people who don't have symptoms of the disease yet, it can have implications for those individuals and could cause a significant amount of stress. So we also want to review the family and the medical history. We also want to assess the risk to the individual wanting to find out their results. We want to provide them with education regarding the clinical and genetic aspects of not only FTD, but also Alzheimer's disease. We want to discuss the benefits, risks, and the limitations of genetic testing. Once again, I want to highlight that there could be psychological implications to a patient and their family members, social implications such as ability to ascertain insurance or to carry on with their job. And also there are implications beyond the individual to other family members. You have to discuss the medical and advanced planning options based on possible test outcomes. We want to ensure that family members and patients have support in making the decision to find out their results. We also want to link patients and families with resources. I think I also want to highlight another important aspect of these genetic forms of disease where there's a very strong inheritance pattern. Sometimes children of individuals who are affected with these genetic mutations that cause FTD or Alzheimer's disease want to start to plan to have a family. And now via in vitro fertilization techniques and embryo selection, you can go through a process whereby, well, without even finding out your genetic result, they can select for the embryos that do not have the mutations, and thereby go through the in vitro fertilization process with certainty that you won't pass on the gene defect even if you have the mutation to your offspring. So another reason to consider genetic counseling, and how with medical technology, things that weren't possible many years ago are possible. I'm coming to the end of my talk, and I'm happy to answer questions, but I just want to highlight once again that there are many different forms of Alzheimer's disease. And here I was talking mainly about Alzheimer's disease due to frontotemporal Alzheimer's disease, and also frontotemporal dementia due to TDP pathology, and frontotemporal dementia due to tau pathology. But there are other forms of dementia as well on this slide that I won't cover. Once again, I want to highlight that there are certain causative gene mutations, particularly presenilin-1, presenilin-2, and APP, that if you inherit one bad copy from one of your parents, you will get Alzheimer's disease. And then there are genetic risk factors, such as the ApoE4 allele. If you inherit this genetic variant, then it increases your risk, but doesn't guarantee you will get it. In frontotemporal dementia, the main genes that cause TDP pathology are granulin and C9orf72. And in frontotemporal dementia due to tau, it's the MAPT mutations. So in conclusion, there are three major genes causing familial Alzheimer's disease: amyloid precursor protein, APP, presenilin-1, and presenilin-2. ApoE is a major risk factor for late onset Alzheimer's disease. And if you inherit two copies of this gene, then your risk for developing the disease can start to approach the risk conferred by one of these stronger autosomal dominant genetic mutations. For frontotemporal dementia, there are three major genes that can cause disease: MAPT, progranulin, and C9orf72. The decision to pursue genetic testing should be made after careful consideration of benefits, risks, and limitations of the testing, and should be done preferably with the help of a clinical genetics team and with family members involved in the process. So I'll end my formal part of the talk there, but just want to describe two exciting research areas that I've been working on for the last many years. So an important area is looking at these rare genetic forms of Alzheimer's disease, which are typically less than 1%. And this is in a study called Dominantly Inherited Alzheimer's Network Trials Unit. And individuals who are able to participate in this gene, in this trial, have to have a known genetic mutation in one of three genes: the APP gene, or amyloid precursor protein, presenilin-1 or presenilin-2 gene. And once again, these are the genes that cause autosomal dominant, strongly genetic forms of Alzheimer's disease. And if you have one of these mutations, you can also be included in the study pre-symptomatically before symptoms develop. And you can also have very early mild stages of the disease, Alzheimer's disease. So this trial offers the potential for treatment during the course of the disease when no symptoms or signs have been demonstrated yet. And as we learn more about these neurodegenerative diseases, targeting them at the pre-symptomatic stage we're moving in that direction, because if we can prevent the onset of the disease, that will be more optimal than treating it once it has started, because once it has started, people already have deficits of the disease. So this particular trial is offering individuals that have genetic forms of Alzheimer's disease, and those who know they have a particular mutation, the chance to be put on what we call open label lecanemab therapy. And lecanemab is one of the anti-amyloid drugs that removes amyloid from the brain that was recently approved in the US. So if you go into this study, you will get anti-amyloid treatment. But on top of that, you also get randomized to either placebo, which is like a sugar pill, or an antibody to tau. So it also tackles the problem of tau. So it's a really unique trial because it's trying to address both of the amyloid issues and the tau issues that we see in Alzheimer's disease. So that's the genetic, this is the DIAN-TU study. And this is typically in individuals who are younger age of onset who have inherited forms of Alzheimer's disease, okay? The information, I think, is presented at the Toronto Dementia Network site. So if people are interested in that, you can reach out there and be put in contact with one of my coordinators. And now I want to describe to you one of the research highlights in genetic FTD. In particular, I want to talk a little bit about the GENFI study or Genetic FTD Initiative. This is just highlighting one of the papers we just recently published, where we looked at blood flow changes in the brain in individuals at risk of genetic FTD. So individuals who participate in GENFI have to have a mutation in one of three genes: the granulin gene, the progranulin gene, the MAPT gene, or the C9orf72 gene. And this study right now is not a drug trial. We're not testing a new agent, but what we're trying to do in GENFI is an international collaboration. We now have over 1,400, 1,500 participants worldwide. It involves large centers in Europe and several other centers in Canada as well. And I'm the lead PI here in Canada, and we provide funding to the other sites to recruit participants for this study. And so what we're trying to do here in GENFI are recruit not only patients that have FTD with genetic causes, but family members. And the idea here is the family members don't have to have symptoms. And also in GENFI, they don't even need to know their genetic results. So if they come in, they only have a 50% chance of inheriting one of these gene defects that occurs in their family member who has frontotemporal dementia. What this allows us to do is using measures such as brain MRI to look at shrinkage patterns, to look at blood flow patterns in the brain, using biomarkers that can be measured in blood, using cognitive testing like pencil and paper tests to assess their cognition and thinking abilities. What we're able to do is we're able to study the natural history of people from the before symptom stage to the after symptom stage. And what that allows us to do is to model how much of a signal to noise we can get, like a signal to noise ratio we can get if we're going to be planning a clinical trial. When we plan a trial and we randomize one set of group of individuals with these mutations to the placebo group and another group to the active drug therapy, what we want to be able to do is determine how much slowing we need to see in these different measures that assess for FTD over time in order to know if a drug is working, while having knowledge of the natural progression rate of these diseases with these particular markers gives us that information. So this study, while it is a natural history study, we are working together closely with our American colleagues as part of the Familial FTD Prevention Initiative to be able to develop clinical trials and design clinical trials using the data we've collected in these natural history studies. We're also working with pharmaceutical companies, and for example, we're running a clinical trial of a drug that can hopefully treat granulin mutation related FTD. So I'm going to stop there. I've said a lot, and I welcome any questions in the last 10 minutes or so. So once again, 2 studies in strongly genetic forms of Alzheimer's disease. We're looking for people who know their mutation status for the APP presenilin 1, presenilin 2, and we have different arms of the study where we include individuals who are 10 years before they're predicted to get symptom onset, individuals who are within 10 years of getting symptoms, and then individuals in the very early stages of disease. In the GENFI study, we're looking for individuals that have these 3 mutations in progranulin, C9 or 72 or MAPT, and we're following the natural history of the disease. And you don't need to know your mutation status to be in this study. So I'll stop there and I welcome any questions.